This note section I refer to as transport in action. It's all applied examples, real-world examples of how transport works in real situations. When you're looking at transport in plants, it really comes down to water and water potential. So water potential, you can see the formula for water potential provided below. There are a variety of sources for potential energy of water. Okay, we, however, will only be looking at two of them, the solute potential and the pressure potential. Why? Well, the mafic and the gravitational do not apply under normal conditions in laboratory settings. They're outside, they're extreme cases, so they are negligible as far as our calculations are concerned. The solute concentration has its own subformula. The solute concentration is equal to the negative I CRT. Both of these formulas are provided to you on your formula reference sheet, and we will look at how to solve them later on in class and breaking down what they are. And I would suggest you take a moment and look at those formulas and make sure that you can understand what each of the letters means. Okay, but there is no formula for pressure potential. So you might have to, in some situations, figure out the pressure potential looking backwards. You can solve for psi s. We can solve for psi p. You sort of have to solve by its absence. So in this example here, if I have a flaccid cell, flaccid cells, psi p is 0. If it's flaccid, there is no pressure from the cell wall. Psi p is 0, so whatever the solute concentration will be the total water potential. Okay, in the plasmolyzed cell, once everything has occurred, I have a water potential of negative 0.9 megapascals. You can get into how you solve that later on as we go. But if you are in a flaccid cell, the pressure potential is 0, or in a plasmolyzed cell, the pressure is 0. Okay, if it is a turgid cell, then the pressure potential will be equal and opposite to the solute potential. Why? Because the turgor pressure pushes back to an equal level so that the cell is balanced. So if it's a in flaccid cell, psi p is 0. If it's a turgid cell, the psi p is equal to the psi s, and the total water potential is 0. So you should be able to figure out that information by looking backwards. So just be aware that you could be expected to do that. Transport in plants depends upon these numbers, and it is something that you are expected to be able to calculate yourself. Okay, outside of that, how does it work? They don't have a heart to pump. Okay, so they depend upon the potential energy of water. Plants are going to export cations. They're going to export these structures. The protons are going to be pumped outwards. Okay, those protons will then be exchanged for protons in the soil. Okay, that will then be brought back in, looking at balancing out those electrical charges. Okay, once that material has entered the cell, it will then flow through a variety of ways through the cells. We can see that there's multiple routes and movements as it goes through the roots and up into the actual vessels, the transport systems of plants. The xylem is what allows water and fluid to move up the cell, and then the phloem is going to allow sugar and sap and all that to move to its storage places. Okay, but the routes within, there are a variety. You have, it can flow through the cell wall completely. It can flow through the plasmodesmata or the symplastic route, or it can go back and forth. Okay, remember the cell wall is not a rigid structure, it's a network of sugars. So we can have lots and lots of transport in a variety of ways through plants. It's really quite diverse. Most plants, though, that is going to be blocked by a waxy area called the Casparian strip. The Casparian strip is outside of these major vessels. Why do plants really need this? Well, if things are just flowing through the cell wall, then they're not being exposed and checked for any viruses. They're not being a sort of immune response. No, they don't have white blood cells, but they do have immune responses. So the Casparian strip forces everything to flow through the cell, at least for a little while, so that immune system techniques can be structured, okay, because that waxy material is impervious to water and dissolved material. Okay, so that's important for the overall structure. Okay, once it reaches the vessels, Once it reaches the xylem, it will use a combination of adhesion, cohesion, and water tension at the stomata to enable evapotranspiration, like we talked about in Unit 1. Okay, this relies upon hydrogen bonding. This relies upon evaporation to power this structure. There is no pump. So this depends upon solar energy and various things transporting water out. Now, that's not always a good thing. Sometimes plants need to stop that water flow. So they do have cells that are on either side of the stoma called guard cells. And by altering the turgor pressure of the guard cells, by making them flaccid or not, it can open and close the stomata and allowing that control mechanism. How do they actually do that? They use potassium ions to alter the water potential and to alter the tonicity. So the flaccid cells are going to go closed, while the turgid cells, the swollen ones, are going to be held open, allowing that flow of material. So it's all, really, when you get down to it, it's a combination of secondary and primary active transport as needed. When you get into more complex organisms, it gets increasingly more complex. Fish, for example, have a lot of transport structures going on in their gills. I know the title says transporting the kidney, but that's really not right. I need to fix that. But they are going to be living in water. So a freshwater fish is going to have a lot more of osmotic water gain because the water is hypotonic to the fish. So they're going to have to excrete a large amount of water, whereas a saltwater fish is going to excrete very, very little water. So you need to look at the type of environment that an organism lives in to try to recognize how water will flow between this. Water is absolutely essential for cellular function. Okay, yes, water can diffuse, but it diffuses very slowly because it has a partial charge. So we even have special transport proteins, passive proteins, called aquaporins, that allow water to move freely. And that's going to be something that we see come up in quite a few places. When you actually look at the actual kidney in animals, it gets more complicated. Okay, the kidney is a series of tubes, which you can see an example of on the right, that is collectively referred to as the nephron. Okay, dozens and dozens and dozens of these structures every few inches inside your kidney. You need to transport waste material out while maintaining as much of the good stuff. Okay, where does the waste material come from? Well, most of it's nitrogenous waste. So when you break down proteins and nucleic acids, you have extra nitrogen and you need to get rid of it. A fish can get rid of it very, very easily as low energy ammonia. But ammonia is incredibly toxic. So if you were to store ammonia in your body, you would poison yourself to death. Okay, animals, we spend energy to make it a safer molecule called urea. Okay, and you're going to store that in your bladder until you pee it out. Okay, birds and reptiles and insects and land snails, they're going to use a more energy wasteful structure called uric acid. But uric acid is non-soluble in water. So when a bird poops on your car, that is not just poop, it's also the urinary waste, the nitrogenous waste, and those white crystals. Why? This typically is something that you see in egg-laying organisms because the egg is going to develop nitrogenous waste as it develops, and you don't want urea inside an egg because it is somewhat soluble in water. So the uric acid is going to build up in the egg without poisoning the young. So very, very helpful there. When you move into the kidney can be incredibly efficient, and you'll see sort of the concentration gradient as you shift. Blood flowing through the body is at about 300 concentration. I honestly cannot remember the units right now. Milliosmoles, sorry, just popped in my brain. Okay, when it comes out, it's 4 times concentrated, about 1200. How do we do that? Well, whenever it goes down on either side, you'll notice it gets more and more concentrated. How? It's pulling out more and more water and salt and that kind of thing. The blue areas are passive transport. The red ones are active transport. As it ascends, it gets less concentrated because the water is going to flow in and the salts and nutrients are going to flow out. Okay, so we're going to be harvesting as we move up, and we're going to be getting rid of water and concentrating as we move down. Okay, allowing for a great deal of efficiency. If you were not able to spend ATP to do this process, then you would have to excrete at the level here. So I would need to excrete 4 times as much to balance out these products. So that's not something that I want to do. When you move into the immune system, you're really looking at things like phagocytosis and pinocytosis. The white blood cell is the most common example of a phagocyte. The phagocyte are going to have lysosomes inside that allow for the digestion of the pathogen. Those bits will be released and then used for immune training and that kind of thing. We'll get into more detail in immune system in a later unit if you're interested, but you get the basic idea. When you move into the actual neural structures, you need a couple of basic understandings first. The neuron is a very specialized cell that uses a combination of both electrical and chemical communication, which we'll talk about in a later unit. Signals are going to be picked up by what's called afferent neurons. So the sensory neurons, the afferent neurons, are going to send signals to the brain where it will be integrated, processed, and then it will then have an efferent neuron, a motor control output, allowing the actual change to occur. But it's going to be a combination of both electrical and chemical communication. But when you really break it down, it comes down to concentration gradients. The neuron itself is going to be using sodium-potassium pumps to allow this process. So the neuron was described as a salty banana. You have salt on the outside, sodium and chlorine. We're going to ignore the chlorine for now. And you have potassium on the inside, like a banana. Okay, so as the sodium flows in, we'll begin to generate an action potential or a charge difference that will initiate a nervous system response. The sodium will flow in. Eventually, that will then trigger that change, which will then propagate down. To reset the signal, the potassium will then flow out until there is a balance and the neuron is reset. Now that's very chemically and energy expensive. So we do have adaptations that allow this to occur at a much greater speed. For humans, we use, for many animals, we use what these called these myelin sheaths. So these are special cells called Schwann cells. They're a special type of cell that support the neuron. And they're really, really fatty, which is why they look white. And it's why we also call like the white tissue versus the white matter versus the gray matter within nervous system structures. They're very fatty, and fat does not conduct electricity. So what we're going to be looking at here is what's called saltatory conduction. The action potential will jump between these spaces, these nodes of Ranvier. So we only have to generate this electrical charge every so often. The large myelin will allow it to speed up. Okay, why don't we just myelinate the whole thing? We need to regenerate the charge every so often or it'll just fizzle out and you'll have no system recognition at all. Okay, how does it actually work on a chemical level? Because that's what we're looking at right now. It's really a combination of these transport mechanisms. So the sodium channels will open first. So sodium is a positively charged molecule. We start off with a negative charge inside the cell. The membrane potential is like, let's just say negative 75-ish. Okay, signal is then going to be received by some sort of receptor. Light hits your eye. I don't know. Okay, once enough light has hit your eye, you will then depolarize. So that will then depolarize. It's going to be moving towards 0, so depolarization, making it less polar. Okay, once it hits threshold, that's our all or nothing signal. If it doesn't hit threshold, it's just going to fizzle out and nothing will happen. But if it does hit threshold, then the sodium gates will open and the sodium will begin to flow in, increasing the positive charge. Once it passes 0, once it becomes more positive, we begin to put the brakes on that process to reverse it. So sodium flows in on this side in step 3. Okay, when it reaches its peak, when the potassium then cancels it out by flowing out. So potassium is flowing out as we drop. Okay, potassium gates close really, really slowly. So we will typically hit sort of an undershoot. It's going to go below where it needs to go. And we're going to need to use active transport to move that back and forth. We're not spending ATP through here. So this is not active transport. If every single steps of this required ATP, your brain would be way too energetically expensive. It really is only small steps that require ATP. So that's really beneficial for our efficiency. Now, not all transport is electrical here, or sort of looking at the ion exchange, we do engage in things like vesicular transport and bulk transport. That's how your neurotransmitters like acetylcholine and dopamine and serotonin, that's how they get out of the cell. The action potential will generate the calcium channels to open. The calcium will trigger motor proteins. The motor proteins will move the vesicle, it will flow out, and it will then bind, creating the next action potential. We're not going into a super great amount of detail here, but you should get the idea. Okay, so this is really, you'll notice that all of those things come together throughout the different types of cells. And all of those ion movements and gradients, it's really how all these things work. Okay, so marine bony fishes, the ion gradients drives the salt excretion. Okay, ion gradients and those things control neuron opening and muscle contraction. Ion gradients control stomata guarding and water release. Ion gradients control ATP production and motor proteins and flagella. All of these things, when you really get down to it, all of these cellular life functions depend upon these concentration and movements. So it's absolutely essential to understand.